Hi everyone, welcome to the fourth lecture in the Immunology series. Now this lecture you have a little bit change of scenery. Anyhow, we're going to talk about the details of the complement system. Now we're also going to look at the mechanisms and steps involved in activating the system and also some selected examples of how it is being regulated. So there is a lot of information in this lecture. Let's get started. All right, here again, some of the lecture objectives. We're going to describe and define opsonizations and also look at the components and functions of major elements of the complement proteins, and also describe the alternative pathway and how it is regulated, the lectin or mannose-binding pathway, and the classical pathway. Let's begin our talk by first defining what is complement. The complement is a family of soluble proteins that are activated during the course of an infection. These complement proteins are soluble proteins secreted by the liver, and there are a family of about 30 proteins, and they are circulating or freely soluble in the circulatory system, lymph, and extracellular fluid. They are considered as something we call cymogens, which are activated by a sequential proteolytic cleavage into an active form. So we're going to look at what it means, and basically that is the central theme and central mechanism of this lecture. Now here as a side note here, I have two words, complement versus complement. Now my accent prohibited me to differentiate the two words, complement versus complement. But anyhow, in this lecture, we are referring to the complement with a E, all right? So what are the basic functions of three main functions of the complement systems? Now, once it is activated, eventually it will lead to three main outcomes. One is opsonization, meaning coating a pathogen to facilitate phagocytosis. Second is inflammation and chemotaxis. Oftentimes we put it together. Now, inflammation promotes chemotaxis, and chemotaxis allows the migrations of different white blood cells to the site of infection. And the third function is lyse, or provide lysis on microbial membranes, which creates holes on the pathogen membranes, and all three mechanisms is going to lead to the death of the pathogen. Now we're going to talk about the complement proteins and systems, and you're going to see a lot of complement proteins with a C. And normally when you are looking at other lectures or other videos on complement, they don't do too much explanations of the naming or the nomenclature of these proteins, and we're going to spend some time to do some explanation so that we are all on the same page. Now, complement proteins start with a C, okay? It stands for complement, and usually there's a number followed, C1, C2, C3, and the numbers are numbered according to the order of discovery and not necessarily the sequence of activation. And for example, in the classical sequence of activation, it goes from C1, 4, 2, 3, 5, 6, 7, 8, 9. So that is out of the traditional numbering order. You're also going to see a lot of letters associated with these complement proteins. And when you see a lowercase, it designates a fragment of the complement proteins. And A means a smaller fragment. A small letter B means a larger fragment. Now, for example, C3A is the smaller product of the proteolytic cleavage of C3, while C3B is the larger fragment. Now, you're also going to see some proteins with just a big letter, a big or uppercase letter, such as uppercase B, uppercase D. These are usually regulatory factors, either activating or suppressing the action of the complement proteins. Now also note that these regulatory factors can be cleaved or can be cut into different fragments. And here is an example. Uppercase B and lowercase b equals larger fragment of the factor B. So keep that in mind. If you get confused with all these different names in the later slides or later half of the lecture, these 2 slides are your go-to lectures, okay? So when we are talking about complement activation, there are actually 3 ways or 3 pathways to do it. And first is the classical pathway. The classical pathway is initiated by antibodies or C-reactive protein, and it has crossed over to the adaptive immune system. It is the first to discover, that's why it was called classical. However, it is the third to act or respond. Another pathway, we call it Lectin pathway or mannose-binding pathway. It is initiated by mannose-binding lectin. It is the second to respond. And actually, the first to respond is the alternative pathway, which is initiating by complement proteins, particularly by C3 binding to the bacterial surface. Now, it is discovered after the classical pathway. However, it is the first to respond. Now, all these pathways lead to a central theme or central enzymes to start the cascade of action, and it leads to the cleavage of C3 to C3A and C3B. And also then the C3b will bind to the surface of the pathogen, leading to 3 main effects. Now, these central enzyme or complex of enzyme, we call it C3 convertase. Now, here is a brief overview. We are going to look at all of these in greater detail later, so hold that thought there. So what are the 3 main effects or 3 main outcomes of this cascade of actions? Number 1, again, we are reviewing. Number 1 is the inflammatory phagocytosis recruitment or phagocyte recruitment, chemotaxis, opsonization, and lysis of the cell or of the pathogen or other structure or membrane of the pathogen via the membrane attack complex or ADMEC. All 3 of these outcomes will lead to the destruction of the pathogen. Let's begin our talk with the alternative pathway, which is the innate mechanism of the complement activation. It is also the first to respond. So we're going to spend a little bit more time in the alternative pathway because once you get a hang of these sequence of activation, the rest of those activations are actually very similar to this alternative pathway. So we're going to go through it step by step. Step 1. Now we would need to have some C3b, right? Remember, C3b binding to the surface of the pathogen is what leading to the cascades of action for destruction of the pathogen. So how do we get some C3b? Well, it turned out that it is initiated by a small amount of hydrolyzed C3 in the serum. Now, the next question is, how do we get hydrolyzed C3? So it turned out that a small amount of C3 is going to be spontaneously hydrolyzed into its hydrolyzed form, meaning that is the IC3. Now this IC3 is going to bind to our first regulatory factor, factor B. Now factor B, and together with the IC3 here shown on the figure, is going to be cut or cleaved by a factor called factor D. Now, you can think of it like a Pac-Man, okay? It's going to chop it up, and mainly it's chop up the factor B into 2 fragments, factor big B, small a, and factor big B, small b. And that big B, small a is going to leave the complex. So the IC3 big B small b complex is actually still floating around, okay? It is not associated with the pathogen yet. It is going to further cut a small amount of C3 into 2 pieces, which is the C3A and C3B. Now, actually, this action, we call it a soluble C3 convertase because this complex is converting C3 into C3A and C3B. Now, after we have obtained our C3b, it is going to covalently bind to the pathogen surface. So it is a very tight, a very strong or permanent association with the pathogen surface. Now, after you get your initial C3b, it's going to start a cascade of action. Now, this is by associating with our factor B. Now, so you've seen this very similar, not only that the first step was happening in the circulatory, and now it is happening on the pathogen surface. Now we have factor D to come in, act like a Pac-Man again, to cut our factor B into big B, small a, and big B and small b. So I realize I've been talking about a lot of B, and, well, reminds me of that. After the previous step, we are going to have a complex, a new complex associating on the pathogen surface, which is the C3b complex with big B, small b. Now this complex, we call it membrane-bound form of C3 convertase. Now again, C3 convertase means it's going to convert C3, okay? Well, it is a positive feedback loop and actually amplifies the responses against the pathogen. So it will continuously act on the C3 and cutting more C3b, providing more C3b on the surface of the pathogen. Now, right now, we have a state where there's a lot of C3b covering the surface of the bacteria. Now, C3b is not going to just sit around and do nothing. And actually, it can also form a complex with another regulatory factors, which is actually forming complex with another big B, small b, and C3b. Well, it is a very complex protein or convertase, which convert C5. Now, we'll come back to this, but when we call it a C5 convertase, it means it cleaves C5 into C5a and C5b. Now, hold your thought on your C5b right now. Now, remember, our C3 has been cut into two pieces, C3a and C3b. C3b remain on the surface of the pathogen, and C3a is floating around. And we've just went through another slide that we talked about how C5 got cut into C5a and C5b. And I told you to hold your thought on C5b, and let's look at C5a first. Now together, C3a and C5a can act on the smooth muscle of the vasculature. It actually can create some contraction of the smooth muscle and leading to increases vascular permeability. Now remember, part of the innate immune response is to have some leaky vessel to allow different white blood cells to migrate to the site of infection. So it will also induce mast cells and basophil to degranulate. And it will lead to something called extravasation of both soluble mediators such as complement proteins, histamine, to mount an immune response, and as well as recruiting phagocyte, neutrophils, monocyte, macrophage to the site of infection. So what we've just looked at about C3a and C5a is leading to the process of chemotaxis and inflammation. And well, what about the C3b again? We coat the pathogen surface with a layer of C3b. What is that called? It is called opsonization. And more precisely, it is called complement-mediated opsonization. So what is the purpose of this opsonization? It is to facilitate phagocytosis process by macrophages or neutrophils. Now, in fact, many bacteria have a slime coat of polysaccharide, I call it sugar coat, on their surface that prohibit recognition of the pathogen by phagocytotic cells, and opsonization is required for phagocytic activity against those organisms. Now let's take a graphical look of the complement-mediated opsonization. So remember, we have C3b on the surface of the pathogen. It is going to bind to a complement receptor on phagocyte, which is a CR1 receptor. Now this binding would create an attachment of the pathogen to the surface of the macrophage. Next, the macrophage will begin with their phagocytosis process and engulf the whole complement bacterial complex vesicle into its phagosome. Now remember, in last lecture we talked about how it do phagocytosis with different means. Now is with the complement-mediated opsonization mechanism. Okay, so once it's phagocytosed or bring in the whole complex, the phagosome, like we talked about last time, is going to join with lysosome containing proteolytic enzymes and other reactive oxygen species such as hydrogen peroxide and fuse into a new unit. We call it phagolysosome. When it's fused, the pathogen is being broken down in that little vesicle. Complement actions can be rapidly amplified via the positive feedback loop, and so far we've seen how a cascade of action leading to a lot of C3b covering the pathogen surface. Now this implies we need something to regulate its actions so that it's active against pathogen but not mediating destruction on human cells. And here we're looking at the first regulatory mechanisms or factor, which is called properdin or factor P. Now factor P, overall, the goal is to stabilize C3b on the pathogen surface. Now this factor P is expressed by vertebrates and it will bind or associate with the C3 convertase. Now what is the C3 convertase? Well, look at the figure here. It is the factor big B, small b, and C3b. Okay, so that is our C3 convertase. It will actually stabilize it and be able to convert more C3b on pathogen surface. The second regulation is about degrading C3b on pathogen surface, meaning where it should not be binding. That involve two regulatory factors, factor I and factor H. Now these two factors need to work together. So factor H is actually a cofactor for factor I. Now these two factors associate together and it will bind to C3b associated with sialic acid. Now once that happened, the factor I and H together is going to cut C3b into IC3b, which is inactivated. Now, so what is sialic acid? Coming back to it. Now, actually, it is something that is present on the human surface. Now, in that case, this mechanism will be able to protect our cell surface not to be coated with active C3b, so that our macrophages is not going to mistakenly eat our cell. However, some bacteria can resist some macrophage actions by fooling the complement system. Now, some bacterial species, such as S. aureus, have some sialic acid on their surface, and through this mechanism, it can prevent being covered with a lot of C3b, so that it can evade some macrophages action. The third regulatory mechanisms involved in dedicated system to degrade C3b on human cell surfaces. And this first factor, we call it decay accelerating factor, or DAF. Now it is expressed on all human cell surface membrane. And the idea for DAF is to disrupt C3 convertase that is bound on human membranes. So how does it do? Now look at the figures here. It will complement the thing that I said in the video. Now this DAF would bind to C3b and compete with factor B. Now this process is going to displace these big B, small b to form this convertase complex. Now because of this disruption, C3 convertase will no longer be able to cut more C3 into C3b and coat the human cell surface. We also have another mechanism that can help us to degrade these C3b on our cell surface. And this second mechanism is through a protein called membrane cofactor protein, or MCP. Again, they are expressed on human cells membrane. And just very similar to factor H, MCP is a cofactor for factor I, and it's going to associate with factor I and starting a cutting action, actually making C3 convertase susceptible by cleavage. And here we see the figure and how it associates with factor I and cut C3b into a inactive form, IC3b. So for the past 20 minutes or so, we've looked at the basic mechanisms of opsonization and what leads to inflammation and chemotaxis. Now we're going to look at the third cascade or hallmark of complement activation, which is forming a membrane attack complex, or the MAC. Now this complex will lead to pathogen cell lysis. Well, remember I told you to hold your thought on a small part of C5, C5b earlier on. Well, okay, so just a little bit recap. When we have a C3b complex with another C3b and factor BB, it will turn into C5 convertase. And this C5 convertase is going to cut C5 into C5a and C5b. And just a little bit recap, C5a together with C3a together can lead to these inflammation and chemotaxis actions in the vasculature. So C5b is the main player to initiate the membrane attack complex. And let's look at the mechanisms and steps together. So we begin with C5b, which serves as an anchor for a big assembly to assemble C6, okay, and C7. So C6 and C7 is going to associate with the C5b. Now, a little bit different for C7 is that it has a hydrophobic site which can insert into the lipid bilayer as shown in the bracket of the below figure. After the insertion of the C6, C5b, and C7 complex, our next complement protein, C8, is going to come in play. Now, it provides a second hydrophobic site that is exposed to an insert into the cell membrane, and altogether creating a 4-protein complex, C5b, 6, 7, and 8. Now, this 4-protein complex is going to guide a process called polymerization. Now, as many as about 18 molecules of C9 is going to form a tube-like insertion into the lipid bilayer of the plasma membrane. So it's going to go in one by one by one and by one. So it's doom, 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 if you try to imagine some sound along with it. Now, after it finished the assembly one by one by one, and it's going to almost look like a tube formation, almost like a channel that creating a hole on the pathogen surface. Now, it's going to create some leakage, such as these water or ions and important cellular contents going to leak out from the pathogen. And water may also enter the cells, and the cells will is going to lyse. So we learn about how we have regulatory mechanisms or protective mechanisms to protect ourselves from being opsonized by C3b. And we also have regulatory mechanisms or protective mechanisms to protect ourselves from the membrane attack complex. It is done by having a surface protein marker called CD59 or protectin, which is a good name, right? Protectin. Now, CD59 is going to bind to this C5b678 complex, and by binding to this complex, it prevents the polymerizations of C9 and subsequent pore formation on the cell surface. So that concludes our discussions of the alternative pathway of the complement system, which is mediated by a series of complement protein. Now we're going to look at the second response pathway, the lectin or mannose-binding pathway. So what is lectin? Now, lectin is an acute phase protein, and it is a polypeptide that is soluble in the serum. Now, lectin is a pretty general term. It indicates the ability to bind to carbohydrates or lipid moiety commonly found on the surface of microorganisms such as polysaccharides and mannose. Now, this particular pathway is about a mannose-binding lectin. Now, here is a graphical illustration of the mannose-binding lectin. Now, other than the binding domain or binding area illustrated by the blue color, it also have some serine protease associated on it. It's called mannose-binding lectin-associated serine proteases, or abbreviated as MASP. Now, keep a look of that. We're going to talk about that in later slides. So this mannose-binding lectin, or MBL, specifically bind to mannose residuals on bacteria, fungi, and protozoan, or even viruses. Now, it will create a process such as those that we've just talked about, opsonization process. So instead of opsonizing with our C3b, it is now opsonizing with mannose-binding lectin. Now, this mannose-binding lectin and mannose complex is going to interact with a specific receptors on phagocytes called collectin. So this collectin receptors is going to interact with this whole mannose-binding lectin complex, and this will then subsequently activate our complement system. So let's look at the steps and mechanisms involved in mannose-binding lectin-mediated activation of complement. So this initiation is by mannose-binding lectin binding to the pathogen surface. Now, and then it's going to follow by the activation of the MASP, or the mannose-binding lectin-associated serine protease. Now, this protease is going to provide a cutting action again. It's going to cut both the C4 and C2 into different pieces. So the C4b fragment is going to bind to the C2a fragment, forming the C4b C2a complex. Now again, this complex is going to serve as a different type of C3 convertase. Now, we've seen a lot of convertase. It's providing a very similar or exactly the same function, but the compositions of it is different slightly. Okay, so keep that in mind. Now, this versions of C3 convertase, again, is going to cut or activate C3 and further lead to the triggering of all of the 3 effector branches of the complement cascade. Now, well, notice that in the bracket here is going to cut C3 into C3b and C3a. So C3b, that action is going to continue, okay, and C3a is going to have a role in the chemotaxis and inflammation along with C5a. So all of those subsequent story that we've just talked about in the alternative pathway will fit into this mannose-binding lectin pathway. Moving on to the last pathway in the complement system, which is the classical pathway. Now, notice that I'm drawing some antibodies here. Now, this is mediated by or initiated by antibodies, both IgG and IgM, can start the process. Now we're going to talk more about antibodies when we start talking about in the adaptive immune system. Now let's look at the steps involved in the antibody-mediated activation of the complement cascade. First, we have antibodies bind to antigens on the pathogen surface. Now, either IgM or IgG can perform this function. Now, notice that the figure here shows how those antibodies are bound to antigens on the pathogen surface. And that is going to recruit a different complement protein, complement C1. And this complement C1 is going to associate or bind to the antibody as shown in the lower half of the figure, forming this C1 antibodies complex. This C1 antibody complex is going to cut complement C4 and C2. Now notice that this process is very similar to just what we just talked about in the mannose-binding lectin pathway. And C4 is going to cut into two pieces, C4b and C4a, and C2 as well going to cut into C2a and C2b. And C4b and C2a is going to form this complex. Well, is it a deja vu? Yes, it's the same thing that we've just talked about, forming this C3 convertase. And this C3 convertase, again, is going to activate C3 and starting all those cascade we just talked about again. Now, antibodies also have a second role. It can actually work with complement together, okay, and mediate opsonization. And these two functions actually have synergistic effect, meaning that when they work together, bind on a pathogen surface, it can enhance greatly the interactions between the pathogen and macrophages and further facilitate phagocytosis process and destruction of the pathogen. Lastly, we also have an acute phase protein, C-Reactive Protein, that is also produced by the liver. Now, bacteria activate macrophages to produce interleukin 6 that we talked about in our last lecture. Other than inducing fever, interleukin 6 can also induce liver to produce reactive protein C or C-reactive protein. Now, when this C-reactive protein binds to the pathogen surface, it is going to recognize by the complement C1. Well, again, it is the same mechanism that we've just talked about. Now, instead of having antibodies complexing with C1, now we have C-reactive protein complexing with C1 and cut complement C4 and C2, forming C4b and C2a complex and turning into this C3 convertase. Well, I'm feeling pretty redundant now. Keep talking about the same thing, and C3 convertase is going to activate C3 and triggering all three effector branches of the complement cascade. Just to remind you, what are the three effector branches of the complement cascade? Recruitment of inflammatory cells or inflammation and / chemotaxis, opsonization, all right, a continuation of covering of C3b, all right, and forming of membrane attack complex. Okay. So here is a summary of what we've just talked about in the past 30-some minutes. We begin with alternative pathway and how complement proteins alone to first act and activate the complement system, leading to a coating of C3b on pathogen surface and ultimately lead to the final three outcome or cascade, which is inflammation, chemotaxis, opsonization, and cell lysis through membrane attack complex, and ultimately lead to the destruction of the pathogen. Then we looked at lectin pathway. The process began with mannose-binding lectin and some different complement protein, but ultimately will still lead to the coating of the pathogen surface with the C3b, and everything else is the same compared to the alternative pathway. And lastly, we look at classical pathway and how the process initiated with immunoglobulins or antibodies or C-reactive protein and associating with the C1 complement protein and leading to a different versions of C3 convertase. Again, ultimately lead to C3b covering the pathogen surface and the final 3 outcome. So there's a lot of repetitive process at the end. The main focus here is to notice some of the initial differences and get those steps in the correct order. So that is all for this lecture. And next time we will talk about acute inflammation.